Welcome to the introduction of bioinformatics. Bioinformatics combine computational power of modern computers to solve biological problems. In a simple way, we can say that bioinformatics is a big data analysis for biology. There are various type of biological data which need specific way of processing to get the meaningful outcome. If we broadly classify the biological data, then it will be sequencing data, image data, agricultural data, drug discovery related data, and protein modeling data. However, this is not the global classification of biological data, but can provide an overall understanding about diversity of biological data. In present biological or bioinformatics research, sequencing data are taking more importance than rest of the categories as sequencing data can be utilized by any of the segment of biology and can provide a deeper knowledge about the particular subject. So let's start with sequencing. Sequencing is a method of reading arrangement of DNA or RNA bases which are responsible for all the characteristics of an organism. Depending on the sequencing technology, we can classify sequencing as Sanger sequencing and next generation sequencing or NGS. Again, depending upon the types of study or the molecule, we can classify the NGS into RNA sequencing and DNA sequencing. Under RNA sequencing, again we have three different classes like transcriptome, non-coding, and rip sequencing. In transcriptome, we try to capture the whole transcript of or the coding RNA expressed by a tissue or a cell at a specific stage or the development or in a treatment. In non-coding RNA, we capture the sequence of different non-coding RNAs, and depending upon the study, we try to relate it with different types of biological regulations. RIP sequencing or ribosomal immunoprecipitation sequencing is a new concept and has a limited implementation to capture and sequence the polysome group or of the ribosome which is binding to the mRNA like beads. It is important to know that RNA sequencing, what we are doing, we are not directly sequencing the RNA as it is difficult to perform the sequencing of RNA. Thus, what we are doing, we are making a cDNA or complementary DNA from the given RNA sample and sequence that cDNA. Let's check what we can do with DNA sequencing. In DNA sequencing, we have options like 16S sequencing, metagenome, exome, WGS, ChIP-seq, bisulfite-seq, pangenome, etc. In 16S amplicon sequencing, we amplify 16S region of prokaryotic organism to identify them. It can be used to identify pathogen in human sample, different organism in environmental sample like water, soil, etc. Metagenomic study can reveal unculturable microbial community and their interaction in an environmental sample and have a great implementation to understand human physiology, environmental problems, etc. Exome sequencing focus only on the coding or the exonic part of genome, and it is very important to characterize the genetic disorder, developing targeted therapeutics, precision medicine, etc. WGS stand for whole genome sequencing, which provide in-depth understanding of genomic architecture of an organism. ChIP sequencing stands for chemoimmunoprecipitation sequencing. It is captured the genomic sequences which interacts and bind to the different types of regulatory protein. This study have immense importance in understanding epigenetic regulation of biological system. Bisulfite-seq, on the other hand, it is talking about methylation of the genomic DNA, and it is actually revealing the part of the methylation which is regulated the epigenetic regulation. Pangenome is a part of metagenomic study which work with entire gene set of study population and express how the core population have similarity or the dissimilarity in their genome. We will go through each and every topic one by one in our next session and also learn how to do the analysis. But before this, let's have an overview about the evolution of the sequencing system. Sanger sequencing is the first generation sequencing which provide up to 800 bp long sequence. This sequencing platform uses chain termination method and the fluorescent dye for sequencing target template. The major drawback of this sequencing system is it is not high throughput system. To overcome this high problem of high throughput system, we developed another system. It is called the next generation sequencing, or NGS. The basic of next generation sequencing is it is short read sequencing and it is a high throughput section. For example, a human genome is 3.2 GB of size, and if we want to sequence it through NGS system, we can get whole sequence done within a week or so. But the same thing if we want to do with a Sanger platform, it may take a year or more than that. Thus, if we are looking for the whole genome, transcriptome, or a big metagenome kind of things, then we have to move for next genome sequencing or next generation sequencing, or NGS. However, there is a third kind of sequencing platform presently underdeveloped, or you can say now slowly gaining popularity. And this third type of sequencing systems are trying to capture the goodies of both the worlds. That means it is try to give you a high throughput along with a long reads. But this sequencing platform presently used as a supplementary system along with another sequencing platforms. So let's start a bit more detail about Sanger sequencing, as Sanger sequencing is a basic of all the sequencing system. Now Sanger sequencing is developed by the Frederick Sanger in 1977. Let's see what all we need to start a Sanger sequencing. Well, if one possesses a template or the target DNA which I want to sequence, I need a primer which will help me to amplify my target. Along with that primer, I need few more chemical components like DDNTPs and DNTPs. DDNTPs stand for dideoxyribonucleotides, which is modified base tagged with different fluorescent molecules. These include DDATP, DDGTP, DDCTP, and DDTTP. Incorporation of any of these molecules in an amplified chain of DNA terminate the elongation of the chain during PCR. DNTPs are the naturally occurring chemical component of DNA, which includes DATPs, DGTPs, DCTPs, and DTTPs. Once we mix this chemical in a buffer solution and perform a PCR amplification, we will get some DNA fragment which is stacked with fluorescent molecule, and I will load this PCR amplified product to the Sanger sequencing machine, it will get segregated by their molecular weight and it will look something like this. Now a laser and a detector will capture the fluorescent signal and convert it to bases such C for cytosine, for yellow, T for thiamine, for red, etc. Finally, this result will be converted and stitched, and software will produce a complete sequence and it will look something like this. This is the final result, which is called chromatogram, coming out from the Sanger sequencing. Present sequencing world is dominated by the single platform which is known as Illumina. Now, depending upon the data output, we have several different types of machine which is using the same sequencing chemistry known as the sequencing by synthesis. For example, MySeq platform can generate around 15 GBs of data, whereas the NextSeq platform can generate double amount of data, that is nearly 30 GBs of data. Let's understand the basic way of NGS workflow. Once we have a DNA sample, we will fragment it by using some endonuclease. This part is very important as sequence platform need small bits of DNA to sequence. The fragmented DNA will be attached with predefined primer and barcodes. Here the red colored and blue colored bar are primer and pink colored ones are barcodes. Barcodes are specialized DNA fragments which are needed to identify the fragment after sequencing by system software, which help it to arrange in a proper manner. Once this step is over, we will get some fragment which will appear like this, in a solution, and this solution called library. This solution or the library then loaded to a NGS machine where there is a special type of a glass slide coated with the complementary primers of fragment present in the library. Thus, they will bind to the glass surface and it will look something like this. After binding, the machine will perform a PCR and generate millions and millions of clones of each fragment known as cluster. Once these clusters are generated, the sequencing will start the similar way the Sanger sequencing happen. Sequencing by synthesis is just like Sanger sequencing system, but binding on fluorescent tag is reversible. Thus, after a cycle of base incorporation, then image capturing, the fluorescent molecule are get washed away, and next bit incorporation is allowed to grow a new DNA chain until it completely done the whole length of the template. In addition, the cycle runs simultaneously to produce millions of fragments at a time to achieve the high throughput. This high throughput obtained after the sequencing is called raw data, and it is stored in a raw data file. In our next session, we will learn what is the raw data and what are the files, what is the data quality, and how to check raw data quality, how to filter this raw data, and how to get high quality data from raw data. By completing the next session, you will be able to open and close the raw data file, and you do QC and you understand what is the QC mean and how to filter the raw data and get high quality data. I hope this session has provided you some insight about Sanger sequencing and related technology. We will discuss further our next session. Till then, have a good day and keep learning with us. Thank you.